Thank you all for coming. It's just really nice to see many familiar faces and that. So thank you for taking the time to come. So essentially my research is focused on understanding species distributions. So this is taken from Alfred Russel Wallace's 1895 Island Life, showing distributions of the jays in this particular case. But it's just illustrative of some of the fundamental questions. Why are these species distributed like this? What are the ecological and evolutionary processes that are going to drive or that have driven these species distributions? And of course, the idea is if we can understand these processes a lot better, then we should be able to build better models and have better understanding of where species are actually distributed, which we often don't know, and also how those distributions respond over time to environmental change. Things like one of the topics I'll talk about is climate change. Now, of course, the tools, the data sets that we have these days are extremely different to what Alfred Russel Wallace had to use, but we're still faced with this fundamental set of questions about the processes that drive distributions such as this. We tend to think about three main factors driving distributions. Just broadly speaking, we would talk about the abiotic niche. So variables such as temperature, precipitation, soil type, say salinity in the marine environment that control and drive where species are found. Then the biotic niche. So the species' role within the ecological community, how it responds to other organisms, is another of the key drivers. And then, of course, there's the whole spatial aspect, so movement, species dispersal, species' ability to move around the landscape. And it really understanding the interactions between these different sets of factors, these different spheres of influence, has been the really kind of main core of my research over the last 15 or it's actually 20 years now. And over the last few decades, this theory of ecological niches has become really quite fragmented, and people use terms in different ways. So, for example, as a macroecologist, someone interested in large-scale distributions, primarily will think about the abiotic niche, kind of following the school, for those who are schooled in this, of people like Joseph Grinnell, where it's the abiotic environment that drives distributions. And then at much finer resolution, if you talk to many community ecologists, people from background learning about the likes of Charles Elton, that whole school of thought is much more that the niche is about the biotic community. So one thing that I've worked on in collaboration with colleagues is a monograph that we published a few years ago now, but that really tries to generate a more modern synthesis of niche theory and how it relates to the biotic, the abiotic community, how movement comes into this, and crucially how that's arranged across different ecological scales, which is something that I will come back to. So I can kind of broadly categorize my work into three main areas. One that we can just say is species distribution modeling, so the technicalities, the details of how we build models of species distributions. Another main interest are the impacts of climate change on biodiversity. And then a third area that I want to get onto mentioning today as well is the evolution of endemism. I think I'd like to show that they're all related, but these could be, these are three separate talks that I've given over the last 3 years, so a few years. So what I've tried to do, for better or for worse, is to piece together just a few highlights from these different areas to try and give a bit more of an overview of these different kinds of fields that I'm interested in and have worked on. So I'm going to try and really give 4 main examples from work over the last few years, looking at Arctic greening, extinction risk in North America, looking at a Californian grassland system, and then finish up with one of my other main interests, which is focusing on Madagascar and evolutionary processes there. So let me start with just this generic idea of a species distribution model. It's one of the most widely used methods in ecology these days. It's really a field that's burgeoned over the last 15 or 20 years or so. The bottom line is that for the vast majority of species on this planet, we actually don't know much about where they're distributed. We might have a few occurrence records. We might just have a type specimen. So actually, using models, using data crunching to try and estimate distributions has been a really fruitful and important area of research for a lot of people. So essentially all we're going to do here, and this is just a generic format, is to say that the probability of some species, let's call it I, occurring in some cell X is some function of the abiotic environment. And this is quite key because most of these models, most of this field will focus on the abiotic drivers of distributions. So actually a lot of my work, as I say, over the last couple of decades has been involved in how we can use different methods, different algorithms to get at this function, which is just a very generic format here, often using machine learning approaches. So a lot of work in that area, and also looking at how we can better validate these models. So you build a model, but then how do we actually validate the model? That would make for a somewhat dry talk if I focused on that today, just going through that stuff. So I'm going to somewhat put that aside and focus on some of the applications. And this is really an area of research that a number of us are interested in because it has kind of angles on quite a few different research questions. So things like looking at predicting species invasions, biodiversity discovery, just discovering unknown species, reserve planning, predicting the spread of zoonotic diseases, predicting the impacts of climate change and land use change. The list kind of goes on, but it has a number of us in this field actually, and you'll be seeing that we have experts at UCL and within CBER as well, in such as zoonotic diseases. Kate Jones is here, species invasions, Tim Blackburn, land use and climate change, Tim Newbold, Georgina Mason, and others. The list goes on. That's just some examples. But this is an area that quite a few of us are interested in. I wanted actually also to just very briefly mention the teaching aspect of this, because, you know, the kind of promotion, if you like, to Professor is not just about the research. This is a research talk, but the teaching is fundamental to that, and this has been one of the areas that I've been put a lot of effort into, frankly, over the last decade or so, to just training people in some of these methods. It's underpinned a lot of my teaching, putting talks out there, making them available, and teaching resources. What I'm going to focus on today is really just one of these main areas of interest, predicting the impacts of climate change, which is something that I have reviewed at book length as well. So let's kick off with this first kind of example of Arctic greening. This is some data from a colleague that I worked with Scott Getz from the Woods Hole Research Institute in the US. Obviously, looking down on the pole, it's based on remote sensing information, AVHRR and MODIS data. But what you're essentially seeing there, the areas in green, are areas that over the last few decades have really increased their photosynthetic activity. And this is exactly what you'd expect to see. The climate has been warming in the Arctic. It's been one of the areas with most pronounced warming. Plants kind of like warmer, longer growing seasons. And that's exactly what we see. So we expect to see plants flourishing in warmer temperatures. We see that in the observed record. In terms of predicting future impacts, this is a particularly interesting study system. Firstly, because, of course, there's some really unique biodiversity in the Arctic. I'll come on to that. Another point of interest is that the biosphere in the Arctic has a really important influence on the global climate system. Again, I'll come back to that and show that. But this is an area of the world where the biosphere interacts very closely with the atmosphere and has a global influence. It's also a part of the world where there's a really strong association, a strong correlation between where you find particular organisms, particular plants, and the abiotic environment. So applying these kinds of methods that statistically associate the abiotic environment with classes of, for example, plants, it's a really good system to be working with. So again, I'm going to skip through, in the interests of getting through a number of examples, I'm going to skip through some of the key methods here, but we use multi-class species distribution models. We run a whole bunch of different climate scenarios. We test lots of different algorithms for doing this. We test lots of different general circulation models, so different climate models. This is just one example kind of output. So this is from remote sensing information, a derived set of categories of where these different classes of vegetation are. So the graminoids, the grasses, 4 different classes. Don't need to go into the detail. The prostrate shrubs, the erect shrubs, so the lower lying shrubs, the taller shrubs, and then trees. So we know where they're currently distributed. We build these statistical models, and then we can project under a set of future scenarios what is likely to happen to those distributions. And overall, we get some quite startling kinds of predictions here. I'm showing you the most extreme example for the sake of giving you a visual interpretation, things that you can see. You can see obviously a very large expansion of the trees under this particular scenario. But overall, across lots of different simulations, the models were predicting that at least half of these areas, half of the vegetated areas would actually switch to a different physiognomic class of vegetation under these future scenarios, with in general larger shrubs expanding while the smaller shrubs lose area. So we can come up with these predictions of changes in different types of vegetation across the Arctic moving forward. One of the key interests here, as I mentioned, was to try and see if we can put some information into, like the IPCC, to actually focus on what the different feedbacks are that are associated with these vegetation shifts. So we can basically take those changes, the model changes in area for each class. We can combine that with remote sensing information that quantifies the radiative forcing, that's the kind of energy exchange for each vegetation class, and from that calculate for each of these scenarios changes for 3 key feedbacks to the climate system. So one of those is a change in albedo. So this is basically the reflectivity of the planet. So ice has a really high albedo. It reflects most of the radiation back into space, whereas a tropical rainforest canopy will absorb most of the radiation. So what we can work out is one of the key feedbacks here is through albedo. It's a positive feedback because the more shrubs you have, the lower the albedo is, which means that more radiation is absorbed by these environments, which means that you get more warming, which means that you get more shrub growth, which means that you get more lower albedo, more radiation. So it's a positive feedback. And we were able to, with very, very broad kind of uncertainty around it, but we could get a ballpark estimate of what the radiative forcing in terms of watts per meter square averaged across the Arctic would be. We could do this for evapotranspiration, ET, up here as well. So this is another positive feedback. The more shrubs you have, the more evapotranspiration. So the more shrubs, the more water vapor is being put up into the atmosphere. Water vapor is a greenhouse gas, so you get more warming, which means more shrubs, which means more evapotranspiration. So again, a positive feedback. And we were able to, by doing this crunching of remote sensing information, we were able to again quantify with big uncertainty, but ballpark the kind of radiative forcing for this increase in evapotranspiration. The other key feedback that had been talked about in the literature, but no one really kind of knew what was the dominant feedback here, is to do with biomass. So of course, again, warming means more shrubs, which means more carbon is stored in the biosphere, which is pulling carbon out of the atmosphere into the biosphere. Less carbon in the atmosphere means less warming, so a negative feedback. So we have these contrasting feedbacks, and what we were basically able to do in this study was to quantify which are the dominant feedbacks here. And the overarching message was that the distribution shifts that we're predicting will result in an overall positive feedback that's likely to accelerate warming, at least in this part of the world. So another angle on this is to think about extinction risk, being one of my main areas of research. So sticking with those same predictions, these kinds of changes in area for each of the vegetation classes, what you're seeing here is change in area. So anything below the 0 line is a loss of area, anything above the 0 line is a gain in area, and then each of these bars is one of those vegetation classes. The different classes of shrubs, trees aren't on here for various reasons, and the different types of grasses. So again, you see some of those classes expand, but many of them are predicted to contract. And the width of these bars is simply because we've got a lot of uncertainty going on here. We're trying to take into account the different assumptions that we make that will give different kinds of predictions. So loss of area of some of these vegetation types, one of the few laws that we have in ecology is that there's a relationship between species, between the area of land that's available and then the number of species that you get. And we can draw on a logarithmic plot, you get a straight line. But essentially, what we're doing then is predicting that less area will mean less species. And this kind of relationship, here's one example from the Arctic, but this kind of relationship has been really widely applied in the literature to infer that under climate change, as certain habitats are shrinking and distributions are shrinking, we would expect to see less species supported. So we infer increased extinction risk. Now, in a system like the Arctic, these shrubs and grasses are the primary producers in these environments. They underpin really quite diverse and unique ecosystems. I've mentioned here Arctic foxes, caribous. I've got in a fuzzy rabbit as well, for those of you who think that us ecologists are mostly interested in the fate of fuzzy rabbits and the like. But the bottom line is then that we're expecting extinction risk as these changes in different vegetation classes that underpin these ecosystems could have knock-on impacts up through the system. And Tom kindly mentioned some of the work that we've been doing with the IPCC, but this is from the research papers. This is then feeding into initiatives such as the IPCC, so actually going from the academic literature right the way through to, we would like to think, having a policy impact. So this is a statement from the fifth assessment report: a large fraction of terrestrial and freshwater species face increased extinction risk under projected climate change during and beyond the 21st century. So that raises quite a conservation challenge, thinking about conservation biology. And there's really quite an urgent need to develop effective vulnerability assessments for evaluating the conservation status of species, particularly under a changing climate. And many of us know about the Iucn Red List. It's widely regarded as the kind of most authoritative system for classifying species according to their extinction risk. Many of us in this room and colleagues throughout UCL and at the Institute of Zoology as well have really been core players in the Red List, its development and its application. But the Red List identifies climate change and severe weather as a threat for about 10% of the 22,000 species that have been listed so far, that have been listed as threatened, which really is there's a mismatch then between this perceived level of threat and what's actually coming through in the conservation assessment. So it raises really two key research questions that have been quite central to some of my work over the last few years. Firstly, what actually is the fraction of species that are threatened by climate change? What truly should we be looking at as this large fraction of species? And secondly, what are the attributes, what are the kind of life history traits, what are the attributes that cause species to have high risk? And the idea being, if we can identify those attributes, then we should be able to do better assessments. We should be able to list species as being threatened better than we currently do. So how do we do this? Again, just very briefly looking at the methods. Well, coming back to my conceptualization of what the different factors are here, we've looked at the abiotic environment. What we at least need to do now is bring in movement. We really need to bring in movement because the extinction risk of a species is clearly going to be very much dependent on its ability to track suitable conditions as the climate changes. So we need to incorporate movement. We need to model population sizes. What I've talked about so far is just changes in suitable areas for species, suitable habitats. If we're going to really have a good go at estimating extinction risk, we need to be saying something about the numbers of species or the abundances. So what we essentially do is say that the population size at time t is some function of the previous population size, the fecundity, so the kind of births, the survival, the deaths, and then immigration, so you add immigration and you take away emigration. So the first part of this is essentially, this is just a classic demographic model. So we model population size against time, where the fecundity and survival, they're density dependent, so you trend up kind of logistic growth up towards some sort of carrying capacity. And then a key thing that we do here is define the carrying capacity as dependent on the abiotic limits. So this is where we use our distribution models that are defined by the abiotic environment to tell us something about the carrying capacity in a particular cell. And the other part of this is movement. Dispersal, it's species moving into, or individuals, sorry, moving into or out of a particular population. So we have a model that estimates abundance, and it incorporates both the abiotic limits and movement. Again, I'm not going to go into the methods in much more detail now, but what we've done, we had a large project when I was based at the American Museum of Natural History, looking at endemic amphibians and reptiles in North America. These are some results for, there's about 35, 36 amphibians and reptiles. So what you're seeing is the extinction risk. This is the likelihood, the probability of the population trending towards zero at 3 different climate change scenarios. A kind of no change. This is encouraging. It suggests we're not just automatically overestimating in our models because there's just a very low extinction risk without climate change. But then a policy scenario, this is a kind of Paris Agreement kind of scenario if we actually mitigate climate change to a degree, and a reference scenario, a bit more of a business as usual type scenario. So what we find with these models is that climate change is going to cause quite a pronounced increase in extinction risk over the next 50 to 100 years or so, looking at 25 to 30% extinction risk, which is broadly consistent with a whole bunch of other studies that have been done using somewhat similar techniques, but different organisms in different parts of the world. So this is the kind of fraction extinction risk that is quite widely coming out of lots of different approaches. Quite interesting to note, not a huge difference between the policy and reference scenario, which can be taken to say, well, the degree of mitigation that we do is relatively unimportant because we're still faced with a large extinction risk, which really emphasizes that we need to be thinking not only about mitigating climate change, but from a conservation of biodiversity perspective, we need to be thinking about ways that we can better adapt, help species, help organisms and communities adapt to climate change. So that kind of leads into this second key question that I've been working with, which is within the context of the Iucn Red List, how can we develop assessments, how can we identify the traits that really make species more or less vulnerable? So because these kinds of methods that we're using incorporate some of the key mechanisms here in terms of extinction risk, what we've done is spend essentially 2 years with some big computers doing huge sensitivity analyses with these models and these bunch of amphibians and reptile species to try and identify for those populations where we are predicting that the population will trend towards zero, that there's high extinction risk, what are the most important traits of the environment, of the species that are driving extinction risk. And it's things that come out are the kind of things that you'd expect, things like population size, occupied area, generation length, number of subpopulations and the like. And essentially what we find, even though there's been a lot of debate in the literature as to whether, particularly the Iucn Red List, whether we really need to rethink the categories that are used, rethink the criteria that are used to define extinction risk, what this research and some related work has really been showing is that most of the variables that we find are actually those that are already incorporated within the main kinds of approaches that we use to assess extinction risk. So in many ways, it's an encouraging story, at least that we don't need fundamentally new approaches, which a lot of people had been saying we needed. And one good side to this from a kind of impact element is that we've worked on guidelines for implementing these kinds of distribution models and these coupled demographic distribution model type approaches through the Climate Change Specialist Group, part of the Iucn Species Survival Commission. They're now written up in the guidelines. There's a long way to go, but at least kind of for the first time, we've got some information in there on how you should implement the Iucn, the red listing criteria, within the context of climate change. So hopefully over time, we're putting out the kind of knowledge and the resources, the tools to increase the ability of people to red list species because specifically of the threat of climate change. Right, so shifting gear a little bit. Everything that I've talked about so far is abiotic environment and movement, but what about this, the big kind of missing factor here so far are biotic interactions, and this has been a real general problem in the whole field that I've been working in for a few years. How can we actually model, how can we get information on biotic interactions into some of the approaches that we use? And we know this is important. We know from empirical studies that this is important. So this is Blake Suttle, colleague of ours who was until fairly recently at Imperial College, who's been working in a Californian grassland system for a couple of decades now. This is his study system. Each of these little circles here is essentially, it's a plot with a pivot at the middle and a kind of old-fashioned sprinkler, adding rainfall, essentially. So what they're doing is an artificial addition of rainfall within this system to mimic some of the scenarios that are expected under future climate change. So what they're really doing is artificial spring watering, so they're shortening the drought season. Mediterranean system, if you shorten the drought system, you would expect the plants to flourish. The abiotic environment should favour these grassland communities, which is exactly what happened at first. So for the first couple of years, the populations became abundant, and the plants, the community as a whole was enjoying it, if you know what I mean. But then actually something really interesting happened after about the first two field seasons. There was a big crash in the abundances of most of the species within this community. And the reason for that is that they were outcompeted by some species, so in particular the annual grasses. So the annual grasses liked the new abiotic environment so much that they were essentially taking over. They were outcompeting particularly the nitrogen-fixing forbs, which are producing the soil facility for many other species. So what happened was, it was the biotic environment that was driving this system, which just illustrates how important the biotic drivers are, and that for most of what we do as macroecologists, we're simply ignoring what is ignoring is too strong, but we're not really fully taking into account these kinds of interactions. So what I'm going to show you is that we've been working with Blake to try and model his study system. But fundamentally, one of the key things that we're trying to do is look across spatial scales and try and link what community ecologists are doing and what us macroecologists are doing. So as a macroecologist, I look at, this is a distribution of one of the annual grasses in this system across the whole of California. The green area here just taken from what's known as the map of life. And it seems reasonable to think that that distribution is in large part driven by the abiotic environment. It's driven by the precipitation and the temperature regimes in this environment. If you Blake Suttle and colleagues looking at just the field scale, what we see is, of course, that it's the biotic environment that's really key. So what we've been doing for a few years now is trying to develop this theory, this conceptual framework where we regard different factors as the driving variables at different spatial scales. So to come up with some framework that can link what's happening abiotically with what's happening biotically. And many of you know Philip Staniksenko here, who was a postdoc in CBER for a couple of years, kind of 2016, 2017. We worked together with Blake and others on this particular system, and thanks really to Philip, developed the mathematical approach for doing this, where we link species distribution models with Bayesian networks to look at the interactions. Now, I'm not going to go through the details of that. I'm going to skip the technicalities, but to give you the broad overview of what we do. Firstly, we take the species in the system. This is focusing on 14 just focal species to get a handle on this. And we use distribution models. We drive macro distributions to drive species distribution models, abiotic environment, to estimate what are the abiotic environment, what's the abiotic niche for each of these species individually. That's the classic. That's what we do within this field. That's what there are tons and tons of studies doing. We treat those as priors, because what we then do is basically wire this system up. So we use the data from the field as best we can. We use the data from the community ecologists' knowledge of what's interacting with what to wire up this system. So what you're seeing around the edge, these species numbers are just, they're just individual species. But within this kind of system, we identify key traits, such as species 34 and 53 are these nitrogen-fixing forbs that facilitate an annual grass. So we use Bayesian networks to wire up this system, and then we model each individual species for its relationship with the abiotic environment, and then bounce this around the network to say, well, if this species is increasing its probability, that's going to have this knock-on impact, which is going to knock-on impact on that, etc. So we're at least, there's a long way to go, but we're at least building a framework where we can start incorporating these kinds of biotic interactions within macroecology and predictions of changes in distributions and abundances. Some pretty pictures of some of the species in this system. Bottom line here is it works. We published a paper in Ecology Letters last year or the year before that basically puts forward this framework and argues that at least we improve our predictive performance. So the y-axis here is a measure of difference between the models without the biotic interactions and the posteriors with the biotic interactions. Anything above the 0 line is an improvement in prediction. Not huge improvements, but improvements nonetheless. So what ultimately, to sum up this kind of area of research, what we're been trying to do is kind of develop a toolbox, a set of methods, so that we can model distributions of species. This is our distribution model, abundances of species, taking into account movement, and then ultimately what we're working towards are whole communities of interacting species, so that we can get at this functional level of how species and how communities respond to environmental change. And I'm not going to go into the details of it now. It's a talk for another time, but we currently are collaborating on a couple of large projects. I see people in the room that are really key involved in these projects, but where ultimately we're interested in how can we better design landscapes to conserve biodiversity and the benefits that biodiversity provides for people. We're looking at 2 study systems, one in South Africa, one in looking at the UK, and we're looking at multifunctional kind of landscapes. So we don't just want to conserve biodiversity, we also need to provide energy for society. So we have a NERC grant, it's what's called the Advent project, which is looking at energy pathways, for example, if we need to grow more biofuels in the UK. And then the Chefs project, which is Wellcome Trust funded, which is looking at how we can develop landscapes that both support biodiversity, but also support the kind of agricultural systems that can provide sustainable and healthy food for society. So there's clear policy links here. If you think 25-year plan for the environment, if you think what's going on politically in terms of managing the landscapes in the UK. But underpinning these kinds of areas of research are the kind of methods that I've been working on. Right, I want to finish up by looking at one of my other main areas of interest, Madagascar. So this is actually work that I really initiated when I moved to the American Museum of Natural History back in 2005. I finished my PhD, which was focused on distribution modeling, extinction risk, plants in Europe, so the kind of stuff that I've been talking about so far, and then took the rather bold move, not just of moving to New York, but of really working in quite a different field. And what I want to show you is why some of these kinds of techniques, looking at niches, have been useful in terms of thinking about endemism and the evolution of endemism. So Madagascar, really fascinating, interesting study system. Big island, it's the world's 4th largest island. Huge degrees of endemism, of course. So if you look at, for example, the amphibians and reptiles, more than 90% of amphibians and reptiles are endemic to Madagascar. Those that are found there, they're not found anywhere else on the planet. But we know why there's high levels of endemism on Madagascar. It's because the island's been isolated from other land masses for upwards of 80 million years. So we know why there's endemism of Madagascar as a whole, but what's much less well understood is why you find these particular patterns of endemism within the island. And you look across lots of different groups. I've mostly been interested in amphibians and reptiles. But here are 3 very closely related day geckos. We link to a lot of phylogenetic work here that have distributions in different parts of the island, and some overlap in distribution as well. But again, it comes back to where I started the whole talk with what are the fundamental ecological and evolutionary processes that are driving these patterns of distribution? Why are some species found in very particular parts of the island? So to go back to some kind of textbook definitions, and there's a lot going on here, but let's first of all just think about the biogeography of the system. So we can first think of what is the classic allopatric mode of speciation. This is hard allopatry, some people will refer to it as. We're going to start with an ancestral population, and there is some geographic divide that causes these two populations to become isolated. It might be a river, it might be a mountain range, but there's some physical geographic barrier that will arise to divide the two populations. It will ultimately lead to isolation. Another area that's been really widely written about in the last 10 or 15 years ago, we might refer to as soft allopatry, so it's another allopatric mode of speciation. But again, you start with some ancestral population, but the idea here is that as the environment is changing, species aren't adapting to different environments, they're tracking known environments, the environments that they're adapted to, which is another way of causing isolation. So a nice way to think about this is to just imagine, and there are different ways, but this is an ancestral population that has two highland areas. As the climate has warmed in the past, which we know it's warmed and cooled many times in the past, then we would expect to see potentially species tracking their suitable conditions to higher elevations and becoming isolated in that way. So again, there's a geographic divide between them that is likely to restrict gene flow and lead to speciation. The two essentially allopatric modes of speciation, but then more controversial in effect is the idea of parapatric speciation. So this is the idea that we have an ancestral population that is distributed along an environmental gradient, and over time populations will become adapted to particular habitats, so subpopulations become adapted to particular local habitats. There's then some sort of assortative mating, reinforcement might operate, but ultimately this would lead to isolation. So this is the idea of clinal speciation. It's something going back to Ronald Fisher in the 1930s, who outlined this theoretically as a potential mode of speciation. We think Theoretically, it can occur, but it's been really hard to identify in nature, and its frequency is really very poorly understood. And yet, if we look at the distributions of many of these amphibians and reptiles, they're often arranged, their current distribution is often they're arranged in bandings along environmental gradients, as I'll mention in a moment. So we can look at present-day patterns of endemism and associate them with these kinds of distributions. So we've done this, and many others have as well, looking across different groups. I've particularly focused on, it's a nice area to work just because of some of these cool critters, such as these just phenomenal Europlatus leaf-tailed geckos, some of the chameleons, some of the snakes. But we can look at the patterns and we can associate them with these different biogeographic modes of speciation. So we find distributions of, we usually look at pairs of sister species, so the most closely related species, and we can find instances where there's a very clear geographic divide that seems to be isolating the populations. We can find cases of high montane endemism, again, where you've got sister pairs that are isolated on mountaintops. But again, what I was saying, crucially, we also very much see in the amphibians and reptiles, particularly elevational gradients, where you get closely related species that just seem to blend into one another across the environmental gradient. So we can come up with kind of scenarios where it seems that the biogeography is pointing towards each of these, or supporting each of these different modes of speciation. But I've spent the last half hour or so talking to you about how distributions shift over time, so we're not going to go and infer too much from the present-day patterns of distribution, but they at least are a good starting point for seeing that whether they at least partially reflect some signal, they show some signal of the original geography of speciation. But really, more powerful, and what I wanted to get onto is to examine the ecological characteristics. So to come up with some ecological hypotheses as what we would expect to see between sister pairs. So here we can think, now we're going to think about abiotic niches, but if this is the ecological suitability along some ecological dimension, it might be temperature, it might be precipitation, going from low to high. Let's actually start down here because it's more straightforward. With parapatric speciation, so each of these curves, I hope you can see the color would be for each of these sister pairs. The whole idea underlying parapatric speciation is that we would expect to see species diverging to occupy particular distinct ecological niches. That underlies the mechanism. So that's a signal that we would expect to see. We refer to it as phylogenetic niche divergence. We expect the niches to distinguish between sister pairs. If you contrast the ecological expectation with this soft allopatry, this is where we would expect fundamentally to see conservatism. So closely related species should have very similar ecological niches, again because the mechanism demands that species are not adapting to different environments as say climate is warming or as the climate is changing, but instead they are maintaining, they're tracking the conditions that they're adapted to and becoming isolated in that way. In terms of hard allopatry, well, this is really a mechanism that's about geography. It's not so much dependent on the niches, so we wouldn't expect to see a statistically significant pattern of trend towards divergence or conservatism in the ecological niches. Might be somewhat similar, they might be somewhat different, but we wouldn't expect to see that they're statistically similar or different. So that's to broadly go over a lot of theory that's been widely written about in terms of niche divergence and niche conservatism. But the key thing is that we can apply some of these similar kinds of ecological kinds of methods to characterize these ecological niches to try and say something of useful in these debates. So this is some really new results, some unpublished work. The method is published, but the results we're writing up at the moment. Again, many of you will recognize Laura Nunez, who did her PhD with us, with me, and she had an impact studentship co-supervised by Mark Wilkinson at the Natural History Museum. And a large part of Laura's thesis was trying to develop these methods for characterizing the similarity and differences between ecological niches. So this is, I think, quite an interesting result. If you look at, so this is a measure of niche overlap. Okay, this is just asking for two populations, how similar are the niches. We look at sister pairs here. We can say that their current distributions are either allopatric or they are parapatric. So what you expect to see is that for allopatric pairs, because they're more distant, we would expect to see that the niches are less similar. There's less overlap in the ecological niches, which is exactly what we see. Parapatric species distributions, because they're much closer together, we would expect to see more overlap in the ecological niches. So that's what you'd expect. But then this is plotting, and there's a long story here, but this is plotting a null model that we published a couple of years ago. We refer to it as the RTR model, but it's a null model that essentially asks, what's the likelihood of seeing a particular degree of niche similarity or conservatism or divergence given the spatial patterns in the distributions? So yes, we expect to see these niches don't necessarily overlap, but within this landscape, based on some null model, what's our expectation for how similar the niche should be? There's a long story there, but we can divide this up then into one axis based on this null model, where we can quantify the degree of niche divergence and the degree of niche similarity, again based on the distributions as they currently are within this particular landscape. And now the interesting thing is that these patterns flip. So now our allopatric distributions we actually find are more similar than we would expect given their distributions in this particular landscape. So they seem to be oriented on the landscape in a particular way where the niches are more conserved than you would expect. By contrast, the parapatric species distributions tend to have niches that are more dissimilar than you would expect given the set of environments in this landscape that are available. So this is, I think, a promising result. It's quite exciting that it's a way of feeding into this broader debate that we can actually not just look at the biogeography, but we can have some reason to look at the ecological niches and again find support. This is showing allopatric distributions do seem to have conserved ecological niches, which points towards an allopatric mode of speciation, whereas potentially this is more support that we are actually seeing parapatric speciation, again because the niches are more diverged than you would expect given the current distributions. So it's supporting expectations, but I'm by no means going to go so far as to say that it's the nail in the coffin that we are actually able to prove these processes. But that leads on to just really where I'm going to finish with, which is to say what we really want to do now is build in gene flow, is to start looking at gene flow here. This is going to give us, again, another part of the puzzle that could be really powerful. This isn't my own area. I have colleagues, particularly at the American Museum of Natural History in New York, who are into crunching the data and working out the methods. But essentially, allopatric mode of speciation, no gene flow. Ancestral population, two diverged populations without gene flow, so an allopatric mode of speciation. If we're going to be able to identify parapatry, then we need to look, in effect, across the genome and identify that speciation divergence has occurred with gene flow. Now, there are methods that are out there to do that, and I know there's expertise in the room in this kind of area. But what's really difficult for us, the key kind of challenge right now, is not just to be able to say, well, we're identifying across the genome that there is speciation with gene flow, but actually we need to be able to identify if it's primary or secondary contact. So we need to be able to identify whether that gene flow has started occurring more recently. They might have diverged in allopatry, shifted around, the distributions have shifted, come back secondary contact, and reinitiated gene flow in effect. So the key challenge out there, and I'm not going to be able to solve it, but I wanted to kind of finish on this, hoping that there might be some collaborators here who can help us think through this. I think there are methods out there, but it's a big challenge. We've kind of got the data, but the challenge is not just can we identify speciation with or without gene flow, but can we go the extra step of identifying whether that gene flow is primary, it occurred at the time of speciation or its secondary contact. And if we can do that, we've got ideas, we're going to shoot a kind of NSF NERC grant. It would be great. I think we could then actually have a bigger picture of piecing together the biogeography, the niche dimension, which is more kind of my area, and then the genomic kind of analyses. So I'd love to chat with anyone if you've got ideas. I will finish there, other than, of course, a whole bunch of people to acknowledge and funders to acknowledge. We've been fortunate to have had a bunch of NSF grants and a couple of NASA grants and others that have funded the work and ongoing funding some of this work. Gosh, having worked for a couple of decades in this field, too many people to put up on one slide that I really need to thank. I've picked up some of the really key people in terms of distribution modelling. So Terry Dawson was my PhD supervisor. Pam Berry, again, colleagues in Oxford, who really just gave me an opportunity to enter this field. At the American Museum of Natural History, particularly Chris Raxworthy was my kind of line manager, my boss, if you like, but a wonderful collaborator. We're still very much in touch, and he is Mr. Madagascar. Eleanor Sterling, who again was director of the Centre for Biodiversity and Conservation, and extremely supportive when I was in New York. The Arctic greening work, particularly Scott Getz, was the key collaborator on this, and Stephen Phillips here, good friend of mine, both across the distribution modelling side and the Arctic greening work as well. We collaborated very closely on that. Rashid Ashokaya and Jess Stanton really were leads on some of the extinction risk, and I've already mentioned Philip Stanek-Zenko and Blake Suttle for the Biotic Interactions work. Just big acknowledgement to CBER as well. This is our picture from the retreat back in 2016. Sorry, I've added a few floating heads for people who aren't on there but really should be. So Laura's here, Fiona Spooner, who recently submitted her thesis looking at some of the demographic modelling work, Henry and Vivian, who are working on the Chefs, the Wellcome Trust project, and of course Georgina Mace as well, who is our PI on that Wellcome Trust grant. I'll say more generally that this is just a terrific place to work, so general thanks out to CBER. I was one of the initial hires, and it's just been wonderful to see how it's developed into this really cool community of lively interacting discussions and people working on related questions. Hopefully you've seen from my link to the Madagascar work, though, that I'm very cautious that we don't want to become compartmentalized into these groups. Love to talk to folks in UGI and Chloe about particularly some of this Madagascar work. More generally, I think all of us within the centre need to thank Georgina, our former director, head of department, director of the division, and the dean for supporting the centre, making it what it is, and they've also supported me personally, so thank you very much. Finally, for those of you who wonder where I disappear off to as of late afternoon, many days, this is kind of where I head. Oops. Wife Sylvina and my two little girls. Key point here, really, it's really important that working at UCL, the flexible working environment, the ability to work core hours and work around that is absolutely vital. So it's another good reason to be here. You'll have noticed that I don't do much field ecology. I am geeky and sit behind the computer, but I still do have ambitions that we might get a Pearson kind of weddy-wearing, if you like, proper field ecologist in future. So thank you very much.